Hello, everyone. Today, it's my pleasure to introduce our work. Systematic characterization of horizontally transferred biosynthetic gene clusters in the human gut microbiota using HTBGC-Finder. The human gut microbiota contains BGCs that encode bioactive secondary metabolites, which play pivotal roles in microbe-microbe and host-microbe interaction. Understanding the horizontal transfer of BGCs can reveal insights into microbial adaptation, resource utilization, and evolutionary mechanisms. Despite its importance, horizontal transfer of BGCs within the gut microbiota remains poorly understood. In this study, we introduce a novel tool, the HTBGC-Finder, designed to systematically identify potential HTBGCs. Introduction of HTBGC-Finder developed a novel pipeline, HTBGC-Finder, to identify potential horizontally transferred BGCs. Identification of HTBGCs discovered 81 potential HTBGCs within the human gut microbiota using HTBGC-Finder. Higher transfer rates. RIPs and CLA BGCs exhibited significantly higher transfer rates compared to non-RPPs and non-CLA BGCs. Phylum-specific transfer. Horizontal transfer of BGCs was detected exclusively within the phyla Bacteroidota and Bacillota. Cross-phylum transfer. Observed instances of cross-phylum horizontal transfer, suggesting a mechanism for rapid evolution within the gut microbiota. HTBGC-Finder is a computational tool designed to identify recently horizontally transferred BGCs within individuals from MAGs. Key components of it include BGCs identification and taxonomy profiling. Construction of GCF network. Identification of outlier BGCs. BGCs identification from reference genomes. Construction of GCFs from MAGs and reference genomes. Phylogenetic analysis. To evaluate the recognition performance of HTBGC-Finder, we applied this tool to detect the potential HTBGCs involved in thiopeptide production in metagenomic samples from the Human Microbiome Project. Among the 1150 bins, 1840 BGCs were clustered into 1035 GCFs, based on similarity. HTBGC-Finder identified the thiopeptide in the dataset and confirmed the possibility of its horizontal transfer by phylogenetic analysis. Following a similar approach, HTBGC-Finder identified a total of 15 potential HTBGCs from the dataset. To further evaluate the performance of HTBGC-Finder, we simulated BGC transfers across different taxonomic levels and observed whether the tool detects the intended transfer events. The ROC curve demonstrated that HTBGC-Finder achieved high sensitivity and specificity, with an AUC of 0.885. This suggests that HTBGC-Finder is highly effective in predicting HTBGCs across different metagenomic datasets. F1 scores for BGC prediction were generally independent of library size, indicating its robustness in handling metagenomes with different sequencing depths. To delve into the intricate phenomenon of horizontal transfer of BGCs within the human gut microbiota, we employed HTBGC-Finder to analyze an extensive dataset from human gut microbiota. As a result, HTBGC-Finder identified 81 BGCs that potentially underwent horizontal transfer in the human gut microbiota. Bacteroidota and Bacillota are the only two phyla where BGC horizontal transfer occurs, with 50 and 31 respectively. RIPs were the most abundant HTBGCs, representing 93.83% of the total. To gain a comprehensive understanding of horizontal transfer phenomena of BGCs, we conducted an analysis of transfer rates. Bacteroidota and Bacillota showed significantly higher BGC horizontally transfer rates compared to other phyla. RIPs displayed a greater transfer frequency than non-RPPs group. CLA-containing BGCs exhibited enhanced transferability relative to non-CLA group. To further investigate potential patterns of BGC horizontal transfer and understand the trends and direction of transfer, we conducted analysis targeting receptors and potential donors of horizontally transferred BGCs. In addition to transfers occurring within the same phylum, we observed instances of potential horizontal transfer between Bacillota and Bacteroidota, as well as transfers from Bifidobacterium and D-sulfovibrio to Bacteroidota. The HTBGC-Finder introduced in this study automates the high-throughput identification of potential HTBGCs within MAGs. Our comprehensive analysis using HTBGC-Finder discovered 81 potential HTBGCs within the human gut microbiota, and RIPs, particularly CLA associated with quorum sensing, exhibited the highest transfer frequency. Furthermore, Bacillota and Bacteroidota were identified as the phyla with the most frequent horizontal transfer events, including cross-phylum exchanges. These findings provide valuable insights into understanding the dynamics of genetic exchange in the gut microbiota, with implications for synthetic biology and drug development.